Hi everyone, welcome to my channel Pure Biology, and today I come with another video, that is the self incompatibility gametophytic. And this gametophytic incompatibility and the sporophytic incompatibility, the two types are seen in the self incompatibility. So its first part is already explained in my channel, so you just check out. Okay, another important thing is that this is explained in my long YouTube channel Pure Biology, and there is another YouTube channel, that is a short YouTube channel, is that Taposhi@education, and you can if you get the short YouTube channel, so there will be the link of the full video of my long YouTube channel in the description box. So you just check out this video from the link. Clear? And there is so come to the explanation. So self incompatibility is what? That is the gametophytic incompatibility, where the S allele that plays an important role. Okay, and in that cases the S allele of the pollen grain, that is coming from the anther, the male reproductive part of the flower, to the to the stigma of the female reproductive part of the flower, when matched there in the S allele in both cases is matched, in that situation the pollen tube begins developing but stops before reaching the micropyle. Clear? So here the pollen tube that is carrying the pollen grain or pollen nucleus, so within the forming a pollen tube towards the ovum, okay, so or ovules you can say. So that is totally stopped before going to the micropyle, that is totally stopped. Okay, and here the two mechanism is seen, and these are the examples, these are the family of the plant, Solanaceae, Rosaceae, Fabaceae. So these are the family of the plants, so where this type of gametophytic incompatibility is seen. So RNase mechanism and S glycoprotein mechanism, the two mechanisms are involved. In case of RNase mechanism, that elongation of the pollen tube that stops within the style, and style is the another part of the female reproductive part of the carpel. So that the determinant genes encode RNase and F box protein. So here the two important proteins are, or the two important factor is very responsible for determining the genes or the locus S, that is the S locus. So two important factors are needed here, and the female component is that ribonuclease, that is the S RNase, and which probably causes a degradation of the rRNA inside the pollen tube. So the two component, one is the male component, male determinant, another is the female determinant. The female determinant here is the RNase, and this is known as the sRNase. And what is the role of this? This is responsible for the degradation of the rRNA that is present within the pollen tube. Clear? What is the role of this? The degradation of rRNA that is present within the pollen tube. Clear? And in case of male, there is the male determinant is that what is that? F box protein. And when this thing is happening, in case of identical male and female S alleles. So this is only possible where the same type of S alleles present in the male and the female gametes. Clear? And so pollen tube, that elongations is needed for the fertilization, that becomes stopped or is arrested. So male component is that the F box protein. Its function is that ubiquitin ligase, that is the ligase activity is occurred by this F box protein, and this functions by recognizing the matching of sRNase molecules. Okay, its function will occur when the total recognition of same type of locus is denoted by the RNase molecules. Clear? And after that, what is happening? Then the proteasomal degradation is occurring. Clear? In the next condition is the S-glycoprotein mechanism. What is happening here? And the female determinant is the small and extracellular and expressed in the stigma. This is present in the stigma, the female determinant that determine the S alleles presence, and the male determinant is the cell membrane receptor. When the interaction between the male and the female determinant that transmit a cellular signal where into the pollen tube, and that resulting the strong influx of calcium cations, and this influx of calcium cations or ions that asserts the tube elongation. Clear? So that means when the two gametes are interacted with each other, a signal or cellular signal that is occurring, and that transmits that enter, you can say enter in the pollen tube. Okay, and due to this calcium cations influx is occurred. Okay, and which asserts the tube elongation. Clear? So the two types of mechanisms are there, and this is the rough diagram where you can see that the stamen S1 and S2, locus S1 and S2, that two are transmitted in two situations. If the S1 is, this is the S1, you can see that this is the S1. When it is transmitted to the stigma, this is the stigma and this is the style and this is the ovary. So when it is transmitted to the stigma, and S1 is not matched here with the carpel S2, because there are the two loci, that is the S2 and S3, so that are not matched by the S1. So you can see that pollen tube elongation is happening. Clear? But in second case, you can see that S2 is transmitted from the anther, and S2 is matched with this S2. So you can see that no pollen tube formation. Clear? So this is the self incompatibility. So you just check out this, and you can read after just seeing or reading this one or understanding this one. You can follow any book, textbook, and where you can get that proper or broad explanation about this self-incompatibility. It's the second part, that is the sporophytic. I will try to explain in my next video. So you just continue with my channel.